All right, so hello, my biochem classmates. Welcome to my presentation. So I'm Michaela, and then the article I chose to present on is called Genome-Wide Kinetics of Nucleosome Turnover Determined by Metabolic Labeling of Histone. So kind of a long title, but we'll get into it. And so this research was done in 2010 by Roger Deal, Georgia Heinekoff, and Stephen Heinekoff, and it was published in the journal Science. So a little background information on the study and kind of what it is. So the purpose of their research was to design a method for determining nucleosome dynamics and turnover across the entire genome. They wanted to study the kinetics of this for nucleosome turnover because these turnovers are super important in epigenome maintenance, gene regulation, and then control of DNA replication. So this has been studied a couple of times before, or at least attempted to, but the mechanisms for these dynamic processes are still not clear, and that's because they're pretty hard to study. So the goal of their research was to see if they could determine an easier technique for measuring the kinetics of nucleosome turnover. So nucleosomes. So we've kind of talked about them in biochem class, and if you've taken genetics or if you're in genetics, you definitely know what they are. But basically, nucleosomes are the first level of DNA compaction. So DNA goes under so many levels of compaction, so it can all fit. And then in nucleosome compaction, basically DNA wraps around a histone protein core, and then this creates kind of a beads on a string look. So that's the picture there that I have on that slide. So it kind of does look like beads on a string. And then together, the nucleosome and histone unit are called chromatin. So the turnover is essentially just the disruption and the replacement of the nucleosome. So the unfolding and then the refolding of the DNA. So you can kind of see why nucleosome turnover would be important for epigenome maintenance and gene regulation and DNA replication. So one previous attempt at studying nucleosome turnover was the mapping the enrichment of the universal histone replacement variant H3.3. So this method required that DNA be completely unwrapped from the histone core in order for the replication-independent histone deposition to occur. So that's what H3.3 does. And then to study this, they measured the steady state levels of H3.3 from fruit fly Schneider cells, HeLa cells, and then cells from, or embryos from a nematode. So basically the steady state levels are when the rate of formation of the intermediate is equal to the rate of its breakdown. So overall, this process took like 2 days, so it was not very efficient. And essentially the results showed that nucleosome replacement occurs across transcribed regions of active genes and at promoters and binding sites that are important for gene expression and then transcriptional regulation. So this research was done by a variety of different people, including the Heinekoffs, who were the ones who worked on this paper, and then it was published in a bunch of different journals as well. So there was another method to measure nucleosome dynamics, but unfortunately this method is only limited to yeast. So basically what it does is it expresses the constitutive genes, which means the genes that are continually expressed, and then it measures, and then it expresses the inducible histone transgenes, and then it measures the relative incorporation of the encoded tagged histones. So essentially, this study showed that turnover rates were high at promoters and chromatin boundary elements, but were low within transcribed regions. So that last part is a little different from what the previous study yielded, but that could just be due to the fact that they used like yeast instead of fruit fly cells. So both of the studies have limitations. So as I said, that last one is only limited to yeast. But they both require transgenes, which are genes from one organisms that are artificially placed into the genome of a different organism. And then they both require tags. And of course, there's a time lag. So it takes like days to complete the process. So Deal and the Heinekoffs wanted to develop a strategy that overcame these obstacles. So their technique is called CATCH-IT, which stands for covalent attachment of tags to capture histones and identify turnover. So what exactly is ketchit? So they use co-translational incorporation of methionine and surrogate azidohomoalanine, which is just an analog of methionine, and that's incorporated into proteins. And then there's the subsequent ligation of an azide group of azidohomoalanine to an alkyne linked to biotin via a 3,2 cycloaddition. So that tagging method is called BondCat, and it was designed back in 2006. So that was kind of confusing. So I do have a picture on the slide of the mechanism. So there you have methionine, and then you have azidohomoalanine, and then the azide group is those 3 nitrogens, and then there it is linked to the biotin linker. And then the steps of ketchit. So to measure the turnover rates, S2 fruit fly cells were starved from methionine, and then they were treated with azidohomoalanine for 3 hours. So then they coupled biotin to the nucleosomes containing the new histones via a cycloaddition. The next step was to purify with streptavidin in order to isolate the biotin tag nucleosomes. So that method is called a pull-down assay, which basically measures protein interactions. Then there was a wash with urea salt solution in order to remove non-histone proteins and other unwanted materials. And then the last step was to analyze the DNA using density tiling microarrays, which is just a tool for measuring gene expression. So I do have a picture at the bottom of the slides of the steps, so just kind of like a visual and a basic summary of them. And then they gathered their data. So the first data they gathered was just a normal ketchit as I described it on the previous slide. So then they did another round of ketchit, but this time after they finished it, they put it back in methionine after the azidohomoalanine treatment. So this was just kind of to see how azidohomoalanine affected the nucleosomes. And then they compared that to H3.3 steady state levels, which is the first approach that I mentioned earlier that took 2 days. And then when all of the arrays were done, they lined up the DNA, and then they took the log2 ratios of pull down DNA to input DNA. So those are the graphs I have here. So the top line, the pink one, is the genes that are expressed the most, and then it kind of decreases. So the blue line, the bottom one there, is the genes that are expressed the least. So when A is compared to B, so A is the normal ketchit method, and then B is ketchit with the methionine chase, you can see that the signals are reduced. So they're just not, they don't have a greater signal, as you can see there. And then they compared A to C. So the C is the H3.3, the steady state levels. And as you can see, the data is actually pretty similar. So that just shows that ketchit did in 3 hours what it took the previous method like 2 days to complete. So then they did scatter plots comparing the correlation between this azidohomoalanine pulse, so just the normal azidohomoalanine treatment, compared to the methionine chase. So that's when they put it back in methionine. And they compared it with all 2.1 million probes versus just the probes, the genic probes from the highest level and then the lowest level of gene expression. So the top line and then the bottom line from the previous slide. So these are showing that nucleosomes undergoing the highest turnover tended to show the largest decrease during the chase, and that depended on gene expression. So if I go back to this, you can see there so they were comparing these two, so obviously there was a greater reduction for the pink ones rather than like the blue. The blue kind of stayed the same. But the ones that were the highest expressed, their signal is the one that reduced the most. And overall, this data just helped determine that ketchit captures the dynamics of histone replacement, and that turnover depended on gene expression level. And then they took more data. So then they observed histone incorporation into chromatin soon after the treatment of azidohomoalanine. So they compared those landscapes of the normal ketchit to steady state H3.3 landscapes. And then they compared it to the binding sites of the landscapes of the binding sites for the ORC2 subunit, which is just a unit of ORC, the origin recognition complex, which just specifies the replication initiation. So it's just an important step in DNA replication. So here's the graph here. So basically what they did is the blue is just the normal catch it. So they removed it at 20 minutes, 40 minutes, and 60 minutes after the azidohomoalanine treatment. And those were all very similar. You can see that. And then when they compared it to the H3.3 steady state level, so that's the green, it was pretty similar, but actually the azidohomoalanine treatment, so the catch it method, had an even better definition of the chromatin. So that shows that catch it depends on nucleosome dynamics and measures turnover kinetics rather than steady state levels, which is what H3.3 method does. And then the ORC2 subunit, so the one in pink, that is similar to both of them. So the ORC2 results kind of piqued their interest with the whole DNA replication thing, so they actually did more data research with that. So they also measured chromatin signals for histone incorporation at ORC binding sites, H3.3 steady state levels, and then different epigenetic regulatory elements. So here are the results. They're kind of confusing, but basically the results from this show that replication origins are determined by chromatin, not by sequence feature. And it also implies that ORC occupies DNA that is made accessible by nucleosome turnover. So that makes sense. So ORC is important in DNA replication. And then we said earlier that nucleosome turnover helps in DNA replication. So overall, catch-it is a great general technique for studying activities that influence nucleosome turnover because it uses native histones and native reagents rather than using transgenes or antibodies as the previous methods did. And then by utilizing this tool, the authors found that nucleosome turnover plays an important role in epigenetic maintenance. So here's some more research that was done with catch-it. So Stephen Heinekoff, so the guy who wrote this paper, and then Sheila Teeves did more research using catch-it. And their research is entitled Heat Shock Reduces Stalled RNA Polymerase II and Nucleosome Turnover Genome-Wide. It was published in Genes in Development in 2011. So this is pretty shortly after catch-it was first invented, and then the whole paper came out. But basically, they used catch-it to measure how heat shock reduces stalled RNA polymerase II and nucleosome turnover genome-wide. So specifically, they used it to test if reduced elongation stage will lead to a decrease in the level of turnover in nucleosomes in fruit flies. And these analyses revealed a decrease in nucleosome turnover for most expressed genes, while some previous silent genes showed increase in turnover. And then Heinekoff actually did some more research in 2013 using catch-it, obviously, with Fan Yang and Christopher Kemp. This research article is entitled Doxorubicin Enhances Nucleosome Turnover Around Promoters and was published in the journal Current Biology. So the goal of this research was to determine if doxorubicin, which is just a commonly used anti-cancer drug, affects chromatin dynamics in cancer cells. So basically what they found was that doxorubicin enhances nucleosome turnover around promoters, and that nucleosome turnover may be a direct result of doxorubicin into DNA. So yeah, so that's all I have for you guys. Any questions and comments you can just leave in the discussion forum on Moodle. But thanks for watching.